Thank you, Stephen. So we do have some time left for questions. We have several in the Q&A box already. So just to remind everyone, please put your questions in the Q&A box in case we run out of time to answer them. The first question is, how do we deal with VUS in rare diseases such as retinal degeneration?
Yep, so that's a good question. Yeah, so it's definitely for rare diseases, especially missense in rare disease, it is more difficult to get those to path or likely path just because there's not as much evidence for those. In those cases, I think a lot of it comes down to a lot of the gene curation aspects that tie into variant curation, like looking at functional domains and knowing, mutational mechanisms to see if there's other criteria that could be applied. But a lot of times those still will end up as a variant of uncertain significance. But I think it's important to remember that someone could still be diagnosed with that disease, even though they don't have a path or likely path variant. That it's important to kind of separate those two components out, that the variant interpretation versus the case interpretation. They still may meet all of the phenotypic features to be able to say they have that disease, even if the variants aren't able to get to a path or likely path level. So I think kind of helping to separate those two out will make it so it's not dependent on a path or likely path variant in order to diagnose those cases.
The next question is, would you advocate for the use of a tool such as VARSM, which automatically applies the ACMG guidelines to help with variant interpretation?
Yeah, I definitely think. Oh, sorry.
Oh, sorry. I was just following on. It says, given, of course, that the investigator critically reviews the output and applies their case-centered knowledge, for example, of segregation data.
Yeah, exactly. So I think that's a really important consideration. Is there are some criteria that could be automated, whereas a lot of the case level ones do require more manual input and decision because it's hard to be able to pull out from a paper exactly how many segregations or de novo occurrences or things like that. For the other types of criteria, I think it depends on sort of a thorough understanding of when that tool is automatically applying the criteria, just so you kind of have a better sense of if you agree with their approach or not. And I think certain criteria types are more easily able to be automated. For example, I think a lot of the allele frequency data, if you've kind of built in those rules that it should be a larger continental population data set, and you know that you want to have at least 2,000 alleles, being able to set rules about when BA1 or BS1 is applied based on that, I think could be helpful. Same with some of the in silico predictors. If you are more confident in Ravel or Polyphen, you could have rules based around automatically applying those. I've also seen, I think it was a tool from BGI called Auto PVS1 that had took the PVS1 flowchart we had developed within the sequence variant interpretation working group and built that into an automated system as well. So I think as we go on, more of these are coming up of different tools that will try to do the automation. So I think it's just important, yes, to understand their rules for the automation and making sure you agree with it. And, some, I think, review of making sure you, look at when it's being applied and see if you agree.
So I have a question. When I was introducing you, I noticed that you are co-leading the ACMG ClinGen working group that's focusing on updating the 2015 ACMG AMP guidelines. And I was wondering if you could just tell us a little bit about kind of what your process is and kind of how that's evolving and shaping up.
Yeah. So we've started this effort, trying to make the next iteration of the sequence variant interpretation guidelines. It's been maybe a year and a half or so ago, and it's still going to be ACMG and AMP, but now ClinGen as a partner as well because of the work we've been doing within ClinGen of working on these specifications. So we have a great group of people working on it. We are trying to move more towards a quantitative point system as opposed to these terms of strong, very strong types of evidence. Because I was kind of showing in the last part of the slides, we think that will help with a lot of the combining rules of being able to kind of figure out when you have conflicting evidence, and also to get a sense of where within a category you fall. For example, like the VUS is a really broad category, that if we know it's between 1 point and 6 points, and if you have a variant that reaches a 5 versus a 1, those are two very different types of variants about where they fall. So I think we are aiming on moving towards more of a point-based system as opposed to these terms. And then as for timing, it's I would say at least a year out still, potentially longer. But we definitely want to do, as we start getting closer and have a better framework to share, of start doing forums with different groups to be able to get feedback, because of course we want whatever guidelines we put out to actually be helpful and useful and adaptable to the community. And so we'll be planning more forums in the future to be able to generate feedback and kind of show what we're working on and get input from people who would actually then be using the guidelines in their laboratories.